Hello and a warm welcome to everyone joining us for today's Select Science webinar. My name is Tamara Reid, Life Science Editor of Select Science, and I will be moderating today's presentation. The topical subject of automation within next-generation sequencing will be discussed today, and we are delighted to introduce today's speaker, Dr. Sean Levy, Faculty Investigator from the Hudson Alpha Institute for Biotechnology, Huntsville in Alabama, USA. I will be shortly handing over to Sean, but to make sure we tailor the webinar content and discussion to you, we would like to take a short poll of 4 questions, the results of which we will discuss at the end. So, when the questions appear on the screen, please select the answer you require and the result will be registered. For question 1, you are able to select multiple answers. So, I see some of you have already started, but I will read out the question. Which of the following do you use to automate your sample preparation? Agilent, Beckman, Caliper, Eppendorf, Hamilton, PerkinElmer, Tecan, or the 2 others? Thank you. I'll read the question again. Which of the following do you use to automate your sample preparation? Okay. Thank you for your responses there. So, for question 2, you may only select one answer here. What is your weekly throughput for sample preparation? Less than 10? Less than 50? Less than 100? Less than 400? And I'll read the question again. What is your weekly throughput for sample preparation? Thank you again for your responses there. So, question 3. Again here, you may select more than one answer. Which of the following do you use for size selection? The Sage Pip and Prep, the Caliper XT-XTE, agarose gel, or none? And the question again, which of the following do you use for size selection? Again, thank you for your answers there. And moving on to the last question. Again, here you may only select one answer. What size ranges do you require? Less than 200, 200 to 300, 300 to 600, 600 plus. And the question again, what size ranges do you require? Thank you very much for your contributions there. Some interesting results. So we will be looking at these after the main presentation. Just before I hand over to Sean, I'd like to mention a couple of things. This is an interactive webinar, and I encourage you to post questions at any time by clicking on the Questions tab at the bottom of the screen. We will aim to address all questions during the question and answer session that will take place after the presentation and the poll results. May I also point out that you can view the presentation in full screen if you wish by clicking on the right-hand tab at the top of the screen. So, without further delay, I'm delighted to hand over to Dr. Sean Levy and want to thank him for presenting to us today. Please go ahead, Sean.
Thanks, Tamara, and thanks to everyone for joining us today, or this morning in some parts of the U.S. And I'll start with a few introductory slides about Hudson Alpha and the operations that we run, and then I'll quickly proceed into some example run formats, and then finally into, I think, what everybody's interested in, some of the automation capabilities and schemes that we've come up with. So the first slide, just an introductory slide giving a simple title for today's presentation: High Throughput Sample Preparation for Genomic Studies. Some contact information for myself as well as the laboratory, and then in the background is a picture of the main building for the Hudson Alpha campus. We're a small nonprofit life science research institute located in Huntsville, Alabama. And then, not surprisingly, since the institute itself is fairly small, myself as well as many of my colleagues here have adjunct appointment at neighboring institutions such as Vanderbilt University or University of Alabama in Birmingham. My laboratory itself is actually split into 4 very overlapping groups, being a genomic services laboratory, which is managed by Melanie Robinson. This operates as a traditional form as a core facility. On the lower right-hand corner, the advanced development group, overseen by Cynthia Hendrickson, is where actually most of the automation development, technology optimization, et cetera, takes place. And then we have research projects as well, shown in the research lab. And then what unifies or centralizes these 3 groups together is an analysis and informatics small group headed up by Braden Boone. I'm going to quickly go through some summaries of operations, and my reasons for doing this is to introduce the type and nature of projects that we run, which I think will help form a frame sort of around our motivations for automation, as well as give you a feel for the types of applications that we're using a few different, or actually using multiple Beckman instruments to currently support. So in the core facility, the Genomic Services Laboratory, as I mentioned, it's a traditional fee-for-service core supporting both projects internal to our institution as well as projects from around the globe. It was founded in August 2009 when I moved my lab from Vanderbilt University here to Hudson Alpha, and since that time we're really kind of officially opening our doors in January 2010. We processed actually now about 49,000 samples over a little over 500 different projects of widely varying complexity, some projects very small, some projects very large. And we're a certified service provider for Illumina, both on sequencing and array technologies, and then certifications for other vendors, Nimblegen, Agilent, Ion Torrent, et cetera, are in progress. The reason I mention our data analysis group on this slide is we feel very strongly that the success of any technology center relies on the ability to appropriately quality control and analyze the data being produced in that center, and that a dedicated, experienced, and efficient group that is very much a central part of our capabilities. And then this allows us to produce software tools in-house, as well as use lots of open source and commercial software. And when appropriate, I'll try to touch on some things there. Advanced Development Group, very similar in reasoning behind the data, similar to the data analysis group, that the cutting-edge nature of most genomic technologies it means that there's often room for innovation and optimization, and then we use this group to kind of actively test, develop protocols for different experimental designs, extend the capabilities, and gain efficiencies, as well as often cost savings on our platform. And then in my research lab, this is really where most of the basic research projects occur, and we certainly leverage our interest in the previously described capabilities, as well as extend them to other scientific areas. Some key projects that I'll touch on a couple of these very briefly. Is whole genome sequencing in bipolar disorder. I'll touch on that primarily. And then exome sequencing in sporadic schizophrenia, variant discovery, and novel and rare diseases, which uses both exome as well as whole genome sequencing. And then some new research efforts, which I'm not going to speak about, is microbiome diversity and its relationship to immune system diversity. And then one final piece that I will actually touch on briefly is the use of ultra-dense barcoding and indexing of samples across sequencing technology. Now, I think this example run batch that I'm showing you here was occurring across multiple HiSeq machines and as well as genome analyzer machines, running a total of 13 flow cells. And the reason I'm showing this is I think reasonably paints a picture as to how we run our operations, combining research interests with development interests with core facility interests, and helps emphasize, I think, what some of the efficiencies that we're able to gain with the automation schemes, and in fact a motivation for having these automation schemes due to the complexities of some of these experiments. So across 13 flow cells, we had a little over 100 pooled sets representing a bit more than 150 individual libraries from multiple investigators, both internal and external, and then again across multiple species with a variety of methodologies being run at the same time: Nextera RNA prep, Agilent Exome, Nimblegen solution-based capture, as well as some array-based capture, Ovation from Nugen Technologies, Illumina's TrueSeq RNA prep, and then 4 different general sample types: RNA-seq, ChIP-seq, exome, small RNA being microRNA, and whole genome. Now, obviously, the sequencing conditions across those 13 flow cells were appropriate for the experimental design. And some of these library preparation procedures were automated and some were not, but all of the quality control, all of the dilutions, and then finally the use of Kappa Biosciences real-time PCR assay for quantitation of library, all of those steps were done on using an automated solutions to generate the final material that went onto the flow cells and through clustering. To more exactly articulate what we have automated and what we use routinely is shown on this slide. So for automated protocols, all sample handling, which generally involves full plate normalizations for concentration, as well as cherry picking samples to go from a received plate into a preparative plate. For example, if we have samples come in from multiple investigators or have multiple investigators, whether they're internal or external, contributing projects to a particular batch, for example, a whole genome sequencing batch or an RNA-seq batch, we'll use the robots to give us a very traceable and trackable solution to consolidate what may be many samples from many plates or tubes into a single plate for the processing. Library preparation methodologies include Illumina's TruSeq DNA prep, Illumina's TruSeq RNA prep, our own GSL, standing for our Genomic Services Laboratory, DNA preparation in a high input form, which means a 2 micrograms of DNA or higher, as well as a form that is a low input form. Sorry, that should say input rather than, or there's a P missing there. A low input form, which is 100 nanograms to 2 micrograms, and then Nugent's Ovation protocol that I mentioned before. On the quality control side, which is certainly more ubiquitous, I mentioned some of these Kappa Biosystems, real-time PCR, final library dilution and pooling, and then finally the pre-cluster denaturation and dilution steps prior to materials going on the Illumina sequencer. So I should emphasize that for the purpose of today's talk, I'll be focusing entirely on Illumina-based sequencing, although we do have some methodologies for ion torrent and actually a very limited number of methodologies for Life Technologies solid platform. But I'll be, for the remainder of today, focusing on the application or these automation schemes specific to Illumina sequencing. But if anyone has, feel free to contact me for anything in addition to what we're presenting today. For sequence capture, we have both the Nimblegen post-Hive processing and the Agilent post-Hive processing automated as well. And some key notes, at least for the specifics of the Beckman deck, is that the tip wash and an orbital shaker, for those of you familiar with the Beckman deck, this will make sense. Tip wash and orbital shaker are required accessories for the schemes that I'm mentioning here, and that actually extra heating positions are not needed for library preparation, or another way to state that is we actually do the enzymatic steps during library preparation with the key exception of PCR at room temperature. We've done the direct comparisons between room temperature or at 30 or 37 degrees, depending on the particular step, and there's absolutely no difference at all in performance. The other key thing is for the sequence capture technologies, we do have some non-Beckman accessories that are needed for these particular schemes, specifically to keep buffers at appropriate temperatures. Those came off of Beckman. So. A final housekeeping piece that I want to be sure I mention, and I have some slides later in the talk which will emphasize this, is we had begun the development of many of these automation schemes prior to Beckman releasing the SPRIWorks HT methodology. Since that's been released, we've had an opportunity to test that, and I'll introduce some of those results as well as some comments on that particular solution a bit later. But I did want to emphasize that everything you see on this current slide was developed or largely developed prior to the SPRIWorks HT. And so we actually find that the SPRIWorks HT and many of these things that I'm showing here are quite complementary to each other, and we routinely interchange between them, and I'll make some further comments on that in some further slides. So why do we automate? Well, it's fairly simple, and I think probably well appreciated by the group. We really use it for accurate and efficient use of the sequencing workflows. We've compared the off-deck as well as on-deck results, and for the same technician doing the off-deck and on-deck, the results are fairly indistinguishable. And that same thing applies across multiple technicians. But where we've really noticed the improvements that the automation brings is when I have where different technicians can use the automated schemes, and we completely, largely, or essentially completely eliminate the bias or differences between different technicians operating the robot, since the robot provides that normalized workflow. Furthermore, it also provides a traceability to the experiments where if something does go wrong, using various tracking features in the Beckman software, you can get a better idea of if something looks funny at the end, it's often a bit easier to trace back compared to manual processes. But I should also emphasize that there certainly are workflows, certainly sample sizes, et cetera, that do not benefit from the automation when you consider the amount of plastics or the amount of time that it takes to set up the robot deck. Excuse me. The automation can be very, very helpful, but in all cases, or it isn't necessarily used in absolutely every case. Two areas that we have found that it is quite indispensable is the automation of the bead purification step, that it can substantially increase both yield as well as consistency. I have a later slide on that. And then finally, as I mentioned earlier, the automation of the library dilution and especially pooling of libraries when indexes are being used and multiple samples are being mixed per sequencing lane. The automation is vital for that as well. So in summary, automation solutions should provide a substantial improvement to both the efficiency of the experiment as well as the quality of data. And that metric is how we determine whether or not we'll actually put additional development effort into a particular automation scheme. So two project examples that I'll touch on that I mentioned earlier: low-depth whole genome sequencing in bipolar disorder type 1, and then just general exome sequencing overall. And in our case, often we use that for recessive diseases in the discovery of de novo mutations. But I think for the purpose of this presentation, it really is general exome sequencing in general. So. A couple pieces of housekeeping information to make sure I remember to mention what protocols, processes, et cetera, are being used in these particular projects. In this case here, we do a standard sample QC, which is just checking integrity of DNA as well as concentration. All of the shearing that we do is done with Covaris to typical size, fragment size, averaging or peaking about 300 base pairs for 300 to 400 base pairs for whole genome libraries, a little bit smaller for whole exome libraries. And then our library preparation methodology, it isn't all of the data that you'll see here is being entirely done with New England Biolabs data. I mentioned the SPRIWorks HT. I'll make further comments on that, but the data you're actually seeing is from reagents from New England Biolabs. It's a modified version of the Illumina adapters. These are not stock adapters, nor are they TrueSeq adapters, but they should be considered equivalent to them, but just for appropriateness, they are a modified version. And then finally, the PCR reagents as well as final library QC reagents is from Kappa Biosystems. On the sequence capture side, it's Roche, Nimblegen V2 and V3. We produce our own blocking reagents from using IDT oligos. Again, Kappa, again, quality control is Pico Green, as well as Caliper's LabChip GX, and then again, Kappa Biosystems as well, and then sequencing on the Illumina HiSeq with varying conditions. So to introduce the project for the low-depth sequencing for experimental design, I won't spend much time on this figure other than to reference a very nice paper from Gonzalo Abacasis and Mike Benke and colleagues at Michigan, which simply illustrates the gain in power that can be achieved for doing very large numbers of samples at low sequencing depth in comparison to smaller numbers of samples at very high sequencing depth. Or stated another way, if you have the opportunity to sequence, if you have a set sequencing budget and you have a large number of available samples, you can gain significant power in a study by doing more samples at lower depth rather than less samples at higher depth for the same sequencing output. And that's essentially what this paper goes into a very nice detail in explaining. And so again, I refer you to the paper for more details, and I only use this as an introduction to that's how we are proceeding forward with a 4,000 sample whole genome sequencing project. Obviously with that number of samples, very high consistency in library preparation and handling is is very, very important. And then sequencing at moderate or low to moderate depth, about a 12x coverage per sample. These samples are indexed, and this slide indicates how they are indexed and pooled using a simple 12 barcode set, which we originally used. We now have a 96 barcode set which are used, but essentially, if you consider a 96-well plate of samples as shown in the upper left corner, each row on the plate using 12 barcodes becomes a single pool, indicating by PS1 all the way through PS8, standing for pooled set 1 through pooled set 8. Now, because the flow cell has 8 lanes, this becomes a very convenient matrix for samples, and that 96 samples get run on a single flow cell. We do a stage 1 flow cell, which is all 96 samples across 8 lanes. Quality control the data, make sure the barcode distribution looks good. If so, then we continue to run that on 8 more flow cells, yielding a total of 9 lanes of sequencing per 12 sample pool, or 9 flow cells per sample plate, and that gives you, if you're averaging 600 or so gigabases per flow cell, or sorry, per HiSeq run, you'll end up in the 12 or 10 to 12x coverage per human genome sample in this particular case. So consistency in library preparation is shown here. This is this was done on the robot deck. In this particular example is a Agilent bioanalyzer. We've subsequently now changed all of this to the Caliper instrument just for improvements in throughput and cost per sample. But the results are essentially indistinguishable. So you see a very minimal to non-existent adapter peaks shown around the 100 base pair line, and then the expected size distribution of each of the samples, highly consistent across this set of 12 samples. Then if we expand that to a plate, you can see row A, B, C, et cetera, are largely indistinguishable from each other, and then this continues obviously through a larger project. So highly consistent, very reproducible library preparation. Using these automated solutions where it's the robot cherry picks the samples, the shearing is done off deck obviously, comes back on deck for end repair through the rest of the library preparation, including all purification steps. There is not a specific size selection step in this particular case other than the standard purifications, and I'll show a slide on that a bit later, and then on to QC sequencing, et cetera. So the consistency is quite good, and the raw mean coverage is shown here for a single representative plate. There is one sample that failed in this particular case. This represents data across a flow cell where one library failed, and so it was removed from sequencing. And then we're seeing between a 6 to 12x coverage, and then percent alignment is highly consistent in the mid 90s, with a few samples that are lower, a bit lower than that. But overall, fairly good consistency across these larger projects. And again, as a reminder, this is whole genome sequencing, only intentionally low coverage data. Some of the sequencing, a nice sequencing quality control step for mixing samples is actually using a very simple ratio of X and Y chromosome coverage. Obviously, a XY ratio in females should be very high, and an XY ratio in males should be much lower. And you can see that represented here, where we have a line drawn at 6, and if any sample that is labeled as male but has a ratio of above 6, we know that sample is cross-contaminated with at least one female sample. And so actually in some of these intermediate male samples, which are shown here, actually represent is a flag for these to be assayed through with other techniques for contamination, whereas everything that's in the higher ratios or the very low ratios generally at least means that you don't have a male-female mixture of samples. And we do intentionally set the plates up during cherry picking to have a fairly equal representation of males and females through the plate, with as close to a male-female alternating pattern as possible, to be able to look for technical mixtures, which fortunately are extraordinarily rare. So stepping away from genome-scale sequencing, which I'm sure everyone's fairly familiar with, this idea that you can take the 3 billion base pairs that represents the human, or roughly 3 billion base pairs that represents the human genome, and sequence the entire genome either at low or higher depth coverage, 30x being I think a common metric, which would represent somewhere in the neighborhood of 90 gigabases of data, or sequence a fraction of the data, say 60 million, 70 million, kind of depending on the version of the exome and the version of the commercially available capture methodology that you'd be interested in, and you can generate a much lower amount of sequence for a higher coverage of the protein coding versions of the genome, which is simply represented in the cartoon shown currently. One of the key things for hybridization-based selection of the regions of the genome is how the library is prepared, and more importantly, how the library is blocked. And this is a basic cartoon which is made intentionally basic to represent whether you're using Agilent or Nimblegen. The results are, or I say the concepts are quite universal in that sense, where you use a hybridization-based solution to target a subregion of the genome. In this case, you're hybridizing for part of the library. You do need to have blocking to prevent these adapter chaining events that can have a specific piece of the genome or a specific piece of DNA that you want represented here, and then over here would be a non-specific piece of the genome that comes along only because of a binding event that occurs between the complementary adapters. So what we found during our automation efforts to develop these methods is that the temperature control as well as mixing is absolutely vital for the efficient sequencing performance, and that post-hybridization, post-hyb automation or making it high throughput is not always particularly straightforward to have on-deck results match results that would be achieved at the bench, and it's mostly due to being absolutely certain you can maintain wash temperatures being vitally important, and then equal or potentially even more important is the mixing steps. And so once the automation schemes are worked out and the testing is done, an advantage of having it automated is being able to very, very routinely or very robustly control those parameters so it is extremely highly consistent from one run to the next to the next. And I'll say that some of our early efforts, as you can see with the tube labeled on the top, shown here, having good specificity on target of a little under 80%. That's actually would represent a bit on the low side nowadays. And then our early efforts with plates and automated, you can see our non-specific was very, very high. And this had to do with actually a very suboptimal control of both temperature and mixing, which has subsequently been resolved very, very well. And I'll illustrate that in a moment. So to summarize a number of experiments, I'll say that on the left-hand side is kind of the very traditional way of doing library prep on individual tubes, capture in individual tubes, and that continues on sequencing. As sequencing output went up and the use of indexing became more popular, it became very routine to do library preparation, again, in individual tubes, capturing those individual samples, but then pooling post-capture material into a single sequencing lane. What I'll illustrate in a moment is our current production methodology, which is library preparation in individual tubes, co-capture with a number of samples, gaining great efficiency both in reagent, but more importantly, in time and consistency, and then continuing on to sequencing. So basically, you're pooling post-library prep, pre-capture, doing a multiplex capture onto sequencing. And so in this case, this slide simply illustrates that pre-capture indexing. If you look shown in, and this is some validation and testing data shown on this side of the slide, samples 1 through 20 here had 1 library per enrichment, pooled after enrichment. And then on the y-axis is percentage of reads mapped to target plus or minus 200 base pairs. So we're in the high 80% to just touching 90%. And then 2 libraries per enrichment pooled before enrichment shown on the right-hand side over here. You can see that these are essentially indistinguishable from each other in terms of performance, or at least in terms of specificity. And that appearance of a potential very slight increase in specificity with the 2 libraries per enrichment, we do see that quite consistently, although I'm not quite prepared to say that it is better than a single library in terms of specificity, but it is at least highly consistent, or it'd be appropriate to say indistinguishable from. When we look at depth of coverage, you see much the same thing. One library per enrichment here on this side of the slide versus two libraries per enrichment here. Again, indistinguishable across this this moderate sample set. And then one question probably on some of your minds is, can you go beyond two libraries per enrichment? The answer is yes. You could actually go to between 4 and 8, depending on the quality of your libraries and the consistency of your libraries. And the one key quality control metric that I would suggest you do if you're considering this process is you absolutely want to use a real-time PCR base, such as a Caliper reagent, prior to pooling before you enrich, and to get a molar equivalent of each of the samples going into the enrichment step. That is absolutely vital. And then, of course, the kappa step, the pooling step, and then the post-hybridization steps for all of these experiments you're looking at here are performed on deck. And this is just reads generated per library, or number of raw reads here, and then 1 and 2 libraries per enrichment. This appears a little bit noisier, but that's because now that we have the variabilities of absolute sequence yield per experiment in this particular case. So, quick highlights as to what does the full process actually get you. So in a quick 45 sample rapid turnaround exome project done with Nimblegen reagents, we had 96% of our clusters passing filter, 92% of the reads were greater than a score of Q30, and a mean quality score of 36 across this sample set, where the entire process was done from library preparation through post-hyb capture. All the quality control steps, dilutions onto the sequencer, et cetera, were done with these automated solutions. So again, just indicating that you can that when each of the links in the chain is well optimized, you can see very nice results with high percentages of and also that extends to getting very efficient yields off of the sequencer, which of course the higher the yields the higher the yield of usable data, then the lower the cost of some of that material. So a couple of other pieces that we found that are quite important, and this is this data summarizes both the effects of good quality control as well as good automation, particularly for library pooling and library dilutions, but in this case it's mostly highlights the library dilutions. We would see this type of cluster variability before comprehensively normalizing the samples. As you can see, indicate here, we have, in this case, a little bit on the low side as far as clusters. It's a bit of an older slide back before the cluster densities on the HiSeqs were recommended to be higher. But we see a range of 400,000 clusters per millimeter all the way down to 200,000, so almost a twofold range shown here, and you can ignore lane 8, which was a failing lane. So as we evaluated all of the key steps for or looked at the variables that went into some of these quantitation solutions and automation solutions, we would often see libraries. In this case, now this is a Caliper GX library trace, where you see this lower marker indicated on the left-hand side. What's labeled as a 100 base pair, very small peak, is a bit of an adapter dimer, and then the library is shown with a fairly robust peak at 292 base pair peak size, and then you see 318, and then you have a tailing off of the library at the end. So this looks like a normal, well-prepared, clean library. And what we found is we also would see libraries that looked like this, where you see the same early steps, but then you see this secondary peak at a much larger size. The challenge here becomes, how do you accurately account for all of the material that is underneath this curve that would extend from here to over here into your quantitation schemes, because the key thing is all of that material actually sequences just fine. The reason for that secondary bubble peak occurs when libraries are over-amplified, most specifically when the primer in the PCR reaction becomes limiting. You end up with a denaturation step during a PCR cycle where then there's no annealing of primer, so instead the complementary adapter ends anneal, and form a bubble product that on whether it be Caliper or whether it be bioanalysis, any of the microfluidic based devices show this secondary peak as a size. If you were to run the same library on an agarose gel, you don't see this bubble product appear. You actually see the single library. The reason the bubble product is so important is if you were to only account for this 295 base pair portion of the peak shown here, you'll under-quantitate or under-represent the amount of your library shown over here, or if you were to use the size of this, the represented size of this aspect of the peak over here, you'll throw off your molar calculations based on that. So the simple solution is you use the 295 base pair peak, or you use the Caliper or bioanalysis only as a size determinant, and then use a real-time PCR as the quantitation determinant along with a fluorescent determination of concentration, and you can begin to see a much more normalized cluster density for library performance shown here. Shown in lane 5 was an intentional overload on this particular instrument. This was when we were evaluating how high we could go on clusters and what effect it had on data yield. So what's shown around 600,000 clusters is a very, very typical performance or consistency between libraries, and you can see a very high percentage of the clusters passing filter and a nice consistency across the flow cell. And if I extend that to what we currently see, for a current project in exome sequencing, that's we see this type of consistency shown here. So now the variability is not, or I should say the consistency is not perfect. There is still some variability due to a number of factors, sequencer consistency included, but we can now very comfortably push cluster densities up to the 700,000 to even 900,000 clusters per millimeter and be confident that we're not going to have lanes vastly overclustered or vastly underclustered. The final thing about library quantitation that I wanted to mention is shown here, which is that we see a very robust performance difference in library type, meaning that Nextera libraries quantitate a bit differently than TrueSeq libraries, which quantitate a bit differently than libraries we make with our with the reagents that I mentioned earlier that were then used in exome capture. As you can see here, that if we were to only use concentration and size, being fluorometric concentration as well as the size of the library, we would see as much as a twofold difference in actual molar amount of the library. When you incorporate real-time PCR to a more comprehensive quantitation, you can then correct for these sorts of differences between these three library types. And this is, as you can see, with a reasonable N number for each one, 51, 18, 88, and 95, and two different TrueSeq RNA batches, and then 2 different exome batches, that it is fairly consistent with each library type. The other thing that we find the real-time PCR is very helpful for is once you have a known library amount and quantitation, we actually use the Kappa real-time PCR results to validate the robotic accuracy. And so in this case, we're testing different replicates using different channels on the Beckman deck, and that essentially as a pipetting step, and we can use this as an assay to determine what type of variability we have. Or if we're developing a new pipetting template, we'll use this PCR methodology to validate the robot accuracy, as you can see here. In this case, this would indicate that one of the channels that's at 36 as well as the other at 49 needs some adjustment. But it's just a simple representation that to introduce the concept of using real-time PCR as a methodology to validate robot accuracy, or at least the pipetting accuracy on the robot, and even be able to use that as measures for looking at different robotic decks versus each other, normalizing one robot. If you have, since we run multiple Beckman decks as well as have a Caliper deck as well, we can kind of use the same assay to look at the pipetting consistency across the different robots. So now, specific to how things look on the Beckman deck, this is actually a deck layout for the Nimblegen method that I mentioned. As you can see, it's not there's quite a bit of there's a number of variables and a number of things represented here. But so this is a methodology that was developed in my lab here on the Beckman deck, and you can see it's a relatively simple layout. This accessory here is this is a non-Beckman accessory. It's simply a plate warmer that has a temperature range of, I think, 40 degrees to 200 degrees Celsius, which allows us to keep buffers at any needed temperature, whether it be 42 degrees, 47 degrees, 65 degrees, depending on which methodology we're doing. And then everything else is fairly standard. And then this is the tip watch station. I did notice there was a question that was asking as to what steps we need the tip washing for. We find the tip wash is just a way to save on plastics and save on tips. By washing the tips in between steps using the tip wash, we can use the same tips more than once. We've done extensive contamination screening for that, and there is absolutely no cross-contamination between tips. But more importantly, the same tip always goes into the same sample, so that it further minimizes the potential negative effect of that. But regardless, it becomes a non-existent issue. It can also be helpful for disposing. It also is very helpful as a waste solution, disposing of materials. And then everything else is again fairly standard on the deck. So I mentioned the SPRIWorks HT, which came along slightly after. This is a representation of its deck, which, again, quite standard. There's nothing overly surprising. This PTC1 on the end that is a thermocycler that is actually not used in the SPRIWorks HT material. We happen to have it on our deck. So this is a representation of the HT deck as configured for the robot deck that we had existing. The only additional accessory here is an orbital shaker, which is at this position here. So the SPRIWorks HT deck is actually a very elegant solution. By using it and having an opportunity to test it, we learned actually quite a bit about or we're able to make improvements to our own methods. And so if you're interested primarily in library preparation, the SPRIWorks HT solution is actually pretty fantastic for a couple of other reasons, which include this type of screen, which allows you to do a number of different things all in a very easy setup. The first being setting the number of samples shown at the top. It doesn't have to be 96. It can be any number in between. Setting the dry time for the bead purification. And then probably something that we found very, very handy compared to our own written methods is the ability to start the library preparation at a chosen step, whether it be end repair, A-tailing, or ligation. You can start at any of those steps and begin forward from there. And I would say most significantly, which this is a feature that is not available on any other platforms that I'm aware of or that I've heard of, is the ability over here to set a size selection parameter, whether it be none, meaning that no size selection on the plate, a small size selection, 150 to 350 base pairs, medium, 250 to 450, and large, 350 to 750 or 700 base pairs. And you can set different wells on the deck to different sizes, all in the same run. So a very, very unique capability for the SPRIWorks HT that we don't have any capability in our own methods, and I think is an exciting opportunity, at least from that, because now you're not switching reagent or switching instrument types to have this size selection capability. And then I think one last slide that is actually very, very convenient, which is once you set those parameters and start a method, it comes up with very specific calculations of how much of each of the Beckman reagents to put in which wells, including how to make the master mixes up. So again, a very, very easy way to have all of the housekeeping pieces or instrument setup pieces, including very exact volumes in both the major reservoirs up here, which is for the ampere reservoir of how much of the ampere beads, how much of the sizing solution, and how much of the elution buffer, but also the enzymatic steps, end repair, the A-tailing mix, the ligation mix, both here and here, and then finally the PCR master mix if you'll be having the robot set up the PCR step, and then again how to make the different, how much of each of the different steps to make, all based on the Beckman reagent. So really, I think hopefully this represents that it's a very convenient start-to-finish solution that certainly has some features that our own scripts don't have. then finally, I think for these automation solutions, when we compare library performance, meaning that so we don't look at things like library yield at any intervening steps or anything else. If we simply take the same sample and put it through our scheme as well as Beckman's, and then put it on a sequencer and look at things such as library complexity, alignment performance, consistency with known sequence data, et cetera. The libraries we've generated on the SPRIWorks HT methodology using their reagents versus the reagents we use, when you get to the sequencing piece, are quite indistinguishable for genomic DNA samples. We have not used it for, I have not done that same comparison for ChIP-seq or RNA-seq or some of the other applications, just to be sure that I represent what data we have correctly. Then finally, I'll skip over this slide. This is simply to indicate that some of you may be aware of this, that there is an error rate that's associated with Illumina indexing. It hovers at around 0.08, 0.09%. Meaning what this means is that the correct index, which is done in a separate read on the Illumina system. So Illumina has 3 reads, which would be the first end read, then the paired read, as well as a third read that reads the index, and then based on that index data, those 3 reads are placed together to identify an individual sample. Well, there's a very small error rate that's associated with that comes from the association of that index read with the correct paired reads. So what we've simply done is developed a slight alteration to the way the barcodes are introduced. That gives us an ability to go up to a little over 9,000 samples per lane with full software support, and more importantly, we can correct for that error. If this is of interest to anyone, in the interest of time, I'll just say please contact me. And then we've actually extended this to kind of what we're calling, for lack of a better term, this Ultraplex 3D, which we can go to actually a little under 900,000 samples per lane. Some of you may be asking, well, that's a ridiculous number of samples per lane. There's no applications for that, but we actually do have a few applications for this that have to do with genome assembly that gets into other advantages of having automated decks for sample preparation. But I'll certainly, we'll discuss those hopefully discuss those at a later time. So with that, I wanted to finish off with a couple things on I think where the automation is really key for, I think, really any number of samples, regardless of what you're starting with. And so whether people in my lab are prepping 6 samples or 600 samples, the purification steps are always done on deck, and for the reasons that you'll see here. The first is that simply changing the ratio of bead solution to sample solution, you can see a fairly crude size selection. I should point out very, very importantly that this is not the same methodology that Beckman is using in their automated size selection step that I showed you in the SPRIWorks HT. This here should most appropriately be considered a relatively crude methodology of doing size selection, but if it gets you the results you need, it actually works quite well. And so this is different batches of ampere beads, as you see here, and then a 0.6x at the top, 0.8x, 1.0x, and 1.8x, where 1.8x being the standard. So, and then the different fragment sizes, and then the value is the percent recovery. So at, I'm sorry, the percent loss in this particular case. So obviously any negative number just means no loss at all, because we're not gaining, we're not making material. It just has to do some of the error in the quantitation at the end. So what this shows is that for any bead ratio, fragment sizes of 25 base pairs probably, or say really anything under 85 base pairs or so, are completely lost. And when you get to 85 base pairs to 100 base pair size fragments, you begin seeing recoveries in the in a reasonable range. And then as you get up to larger fragments, you see essentially no loss at all at these larger sizes. So you can tailor this percentage to get size selections in the 100, say, to 300 base pair range that are pretty efficient. Or arguably what we use it most for is just making sure that whatever adapter design you're using, or adapter plus PCR primer you're using, you can eliminate those products. And then this slide, I think, indicates two things. One is that the purifications can be highly efficient, and secondly, that there are great opportunities with the automated solutions to using very, very low input amounts and retaining that recoverability by doing slight alterations to the bead protocol. So in this case, it is retaining the same aliquot of beads, and then instead of adding new beads for subsequent purifications, actually adding the binding buffer alone. An easy way to do that is you take the same impure beads and just spin the beads out, and then use the binding buffer for the subsequent step. So you're still using the same amount of beads, or really the same amount of reagent for each step, but you do gain a little bit of an efficiency in this step here. And so what you're seeing is different fragment sizes, and then the gray diamond indicates 0 bead cycles, and then the red box, meaning that no purification step, and then the red box indicates the amount of DNA retained for that same fragment after 5 cycles of bead binding, washing, and releasing. So what you can see is for really at 75 base pairs and larger, you're seeing no loss at all across 5 cycles by using that methodology that I mentioned, a single aliquot of beads that start in the beginning. The beads are released with the master mixes of the subsequent library steps, the DNA is released off the beads with the subsequent master mixes of the library steps, and then rebound using an aliquot of binding buffer from the ampure reagents. You see almost no loss at all. I should give credit where credit is due, is that this methodology was introduced, I believe, from Chad Nussbaum's group at the Broad Institute and published in the original Agilent capture paper that came out some time ago. So this idea is not our idea at all, but I did want to present that. So. Finally, and to conclude, here's some recent references that I just wanted to put up for papers that have come out of sequencing data that the lab has produced. And then finally, to give acknowledgement both to my colleagues at Hudson Alpha and some of the places we're collaborating with for this. And then I'll end with the final slide here with a picture of my group, as well as and then the groups from both Ingenuity, Roche, and Beckman, who we've been working closely with on some of this development work. So. With that, I'll thank you for your attention, and we'll move towards the question and answer session.
Thank you very much, Sean. Thanks, that's great. Thank you for the informal presentation. We have some questions for you coming in while you've been presenting. But before we look at those, let's take a look at the poll results. I'm sure you've all been excited to find out the answers. So for the first question, let's see. So it's always interesting to see what platforms are being used. So thank you very much for responding to that question. And moving on to question 2. Wait for it to come on the screen. Great. In terms of weekly throughput, we can see that you are using high volumes in your labs each week. Sean, would you like to comment on these results?
Well, I think really for the full solution, or the full automated solution, really anything 100 samples and above, I think strongly justifies or can strongly benefit from the automation. And I think these poll results clearly indicate that. I think a number of folks are interested in or are using samples at that at that density.
Yeah, great. Yes, thank you for that, Sean. So let's move on to poll number 3. Have a look at the results of that. Again, thank you very much for responding to those answers, and it's always interesting again to see what the majority of people are using in their labs. And so let's move on to question 4. Thank you. And again, question 4. Sean, do you have any comments on this interesting result?
Yeah, I think that's somewhat expected. 200 to 300 base pairs, I think, is typical for Illumina sequencing when you're using paired 100 ranges. One thing I would like to comment very briefly is that we found the size selection ranges on the SPRIWorks HT are actually significantly tighter than we see with other methodologies. And so if the ranges you saw on the slides are of interest for your research and having that peak have very steep sides to the, or a high slope to that peak, looking at the SPRIWorks HT may be of significant benefit.
Lovely. Thank you very much, Sean. So let's move on to the question and answer section. We'll ask you a couple of questions we've received in. So, Sean, the first question we had was, the growing technology of nanopore sequencing does not need any sample preparation. How do you think this will affect lab automation in next-generation sequencing?
Sure. I think that's a good question. I would have a two-part answer. Number one is, so nanopore sequencing has been introduced. It's an exciting technology that'll be here in the future, but it doesn't solve, it is still some time away from solving work that needs to be done today. So specifically in our core facility, I certainly, or even our research laboratories, we couldn't put all projects on hold for nanopore sequencing and wait for that to come out. So that's really, I would say that the automation solutions are available now, for experiments that are being done now. And how technology will change in the future is no different than how it's changed in the past, is that as new opportunities become available, we'll certainly proceed with those opportunities. But the second part is, I think the statement that it doesn't need any sample preparation is, it may be a somewhat an oversimplification of the field. For whole genome sequencing, that would be true, absolutely true, and that you may be able to take naked DNA through the nanopores and get sequence results, perhaps with RNA as well. But applications like reduced representation, methylation sequencing, exome sequencing, et cetera, there still would be a need for sample preparation. So I think even if we assume the best outcome of nanopore sequencing and the most efficient cost ratios, there will absolutely still be specific applications. Now, whether or not they're in the majority or the minority is a debatable point, but still will absolutely be some need for sample preparation, even if it's just the quality control and other aspects that these automation solutions would still be appropriate for. Great.
Thank you very much. Thank you. And we've got another question here regarding the bubble peak in the library. The question is, you mentioned you use qPCR to calculate the actual molarity instead of using the shown size. But if we have to use the size, should we use the smaller size for calculation or a number between the big and the small sizes? Bit of a long question there, Sean.
Yeah, no, actually, I would recommend neither. And if you see the bubble product and you can make a guesstimate and use the smaller size as the fragment size for the molarity calculation, and then use the fluorescent DNA determination. That will get you closest to the correct answer, because the fragments are not actually bigger. It's artifactual that they run bigger, so you still want to use the smaller size. But you can get rid of the bubble product by simply taking that same library, adding in the same amount of primer you went into the original PCR mix with, and obviously with appropriate enzyme, and do 2 cycles of PCR, and it'll collapse that peak down to a single one, because you basically reset all of those products back to their correct product.
Great, thank you. And I've got another question here. What do you think about the SPRIWorks robot? It just works with Beckman library prep kits, so it's a pretty close platform compared to the Mondrian robot.
Yeah, so this we worked with SPRIWorks in the past, and to provide an honest answer, we were neither impressed with, really weren't impressed with the SPRIWorks robot for our workflows. But again, remember that we're approaching it from the perspective of plate-based library preparation and large numbers of libraries. I think it may be a good fit for laboratories looking at relatively small numbers of libraries, and I would only, in comparison to the NuGen robot, it's kind of an apples to oranges comparison. The NuGen robot is very much designed for very, very low input material and very, very small volume reactions, which have its own, I think that if that's a primary consideration, the Mondrian is a good fit. And then if a standard input, kind of standard library preparation solution, just small numbers of samples, then the SPRIWorks may be a good fit. But I think they're similar in terms of sample throughput, but beyond that, I'd consider them actually very, very different instruments with very different target experimental designs.
Great. Thank you very much for that, Sean. So I'll just ask you one final question, as I appreciate we're running out of time now. You mentioned about tip washing steps. I think this question has been more or less answered, but if you'd like to add any more, that would be great. The question is, can you please talk about what steps you would need tip washing for?
Yeah, I think I touched on this before, so I won't spend too much time on it. At this point, it's really just a opportunity to save both time on the robot, or three things. One is it saves some time where you're not changing tips when appropriate. Secondly, it gives you an ability to reuse the same tips to save on plastics consumables. And when I say reuse, I mean reuse within the same method. We actually don't retain tips between methods. And then finally, it is a very convenient way to dispose of waste, where usually the elutions that are removed, I'm sorry, not the elutions, the binding buffer or the wash buffers that are removed and then discarded during the bead purification steps can be discarded in the wash stations because they're rinsed away. And so overall, we find it to be a very advantageous piece to the robot deck, but optional. You certainly have the option to use brand new tips throughout a particular method. There's no limitation there. It's just an opportunity to, when appropriate, and when things like contamination, et cetera, is appropriately controlled, that you can reuse the same tips and save a bit of money on consumables.
Okay, great. Well, thank you ever so much, Sean. That's all we've got time for today. Thank you for your informative discussion and presentation, and thank you to everyone joining us online and for your interesting questions. I hope that you've all found this a worthwhile session. If we didn't manage to answer your questions, we will follow up with you all personally, and if you have any more questions, please feel free to email me at editor@selectscience.net. If you'd like to listen again to today's webinar or invite a friend to listen, it will be available on our website shortly. So, thank you, goodbye, and thank you all once again.